Thank you all for your patience. It appears that most of the participants have joined the conference, so we can officially begin now. First of all, a very good morning to one and all present here. I am Chetna Jhatta, the organizing secretary of the event and assistant professor at Government College of Pharmacy, Rohru. On the behalf of entire Government College of Pharmacy, Rohru, I extend a warm and hearty welcome to all the distinguished guests, speakers, participants, and delegates attending the first international conference on computational drug design, trends, challenges, and future prospects. We are truly honored by your presence and look forward to fruitful discussions and knowledge sharing experiences. It gives me immense pleasure to honor and welcome you all to the first international conference organized by our institute. First of all, I would like to introduce our college. Government College of Pharmacy, Rohru was established in 2005. It is the first government pharmacy institute in the state of Himachal Pradesh dedicated to imparting technical education in the field of pharmacy. The institute offers a 4-year B Pharmacy program and is approved by All India Council for Technical Education as well as Pharmacy Council of India, New Delhi. It is affiliated with Himachal Pradesh Technical University, Hamirpur. We are also very proud to share that we are the first NAC accredited government technical institute in Himachal Pradesh, a milestone that reflects our commitment to academic excellence and quality-based pharmacy education. Today, as we gather here for this prestigious international conference, we embark on a journey to explore the cutting-edge advancements in computational drug discovery, including artificial intelligence, molecular modeling, and machine learning. So this conference brings together renowned experts from academia and industry to discuss emerging trends, challenges, and future prospects in this dynamic field. So the objective of this conference is to foster collaboration, innovation, and knowledge exchange to accelerate drug development and discovery. So before we proceed further, let me take a moment to acknowledge the presence of our esteemed dignitaries. Our Chief Patron, Mr. Akshay Sood Sir, Himachal Pradesh Administrative Services, Director Technical Education, Vocational and Industrial Training, our Patron Director Principal, Government College of Pharmacy, Rohru, Professor Dr. Vivek Sharma Sir, all our esteemed speakers, invited guests, and delegates from India and abroad. Now, before we officially begin, I would like to brief some rules regarding conference etiquettes and guidelines. First of all, keep your microphones on mute during presentation to avoid background noises. In case of any technical difficulties, our technical support team is available to assist you. For question answer session, you may either use raise hand option in the chat box or can unmute yourself one by one to avoid unnecessary disturbances. And 2 feedback links will be shared in the chat box. So please do not forget to fill it out as it is essential for completing the attendance criteria. And I also want to do some important announcement regarding e-poster presentations. Participants who are the part of e-poster presentation are requested to remain active on the separate link handled by Mr. Pankaj Jinta, co-organizing secretary of the event. And the result of e-poster presentation will be declared at the end of conference. So now to commence the inaugural ceremony, I would like to invite our patron, Director Cum Principal, Government College of Pharmacy, Rohru, Professor Dr. Vivek Sharma Sir, to deliver the welcome address. Over to you, sir.
Thank you, Chetna. Thank you so much. Respected Chief Patron Shri Akshay Sood Ji, honorable keynote speakers, revered faculty members of different institutes, distinguished researchers, and dear students. I am deeply honored and privileged to stand before you as a host in the first international conference organized by the Institute on Computational Drug Design. This conference has been organized in collaboration with Department of Technical Education, Government of Himachal Pradesh. And this is a historic occasion that signifies not just a meeting of minds, but a milestone in scientific advancement. This gathering is more than a conference, and it is a witness to the power of human imagination, collaboration, and the persistent search of innovation in the healthcare. We are living in an era where computational drug design is reshaping the foundations of medical science, and which is now no longer confined to trial and error. Drug discovery has entered a new frontier, which is driven by AI, big data, and advanced modeling, which has transformed the speed, accuracy, and the possibilities of pharmaceutical breakthroughs. With these cutting-edge tools, we are not just treating human sufferings, but we are imagining a future where the impossible becomes achievable. Today, we are privileged to have with us stalwarts in the field, our President Awardee, Professor Kumar, Dr. Neha Tripathi, Dr. Kanhaya, and Mr. Akbar Shaikh, whose contributions have redefined the boundaries of computational drug design. Their presence here underscores the importance of collaboration, knowledge sharing, and the collective mission to revolutionize healthcare. I extend my deepest gratitude to the organizing committee for their dedication and hard work in bringing this conference to life. To all the researchers, faculty, students, and industry experts, you are the driving force behind scientific advancement, and your passion, interest, and commitment are the pillars upon which future discoveries will stand. Let this conference be more than an exchange of ideas. Let it be a catalyst for groundbreaking partnerships, a spark for novel innovations, and a platform that shapes the next generation of pharmaceutical breakthroughs. Let us dream of a future where diseases that once seemed insurmountable and conquered, where suffering is replaced with hope, and where science continues to be the greatest gift to humanity. It's my duty to acknowledge that if we have fallen short in any way or have not met your expectations, because it is our maiden effort, first time we are organizing this, because of any technical glitches, I seek your understanding. But your presence here matters a lot. It is valuable, and we are contributing collectively to a moment of significance and progress. So at last, I extend my warm welcome to this transformative event. May it inspire, challenge, and propel us towards a healthier, brighter tomorrow. Have a great day ahead. Thank you so much.
Thank you so much, sir, for your encouraging words and for officially inaugurating the conference. Once again, I extend my warm welcome to all the distinguished guests, delegates, and participants. I am confident that this conference will prove to be a great platform for meaningful discussions and fruitful collaborations. Now, I would like to hand proceedings to our co-organizing secretary, Mrs. Shivani Ma'am, Assistant Professor at GCP Rohru, for further conduction of the event. Shivani Ma'am, are you there?
Yes, ma'am. Let me share the screen first. Is it visible to all?
Yes.
Yes, ma'am.
Now we will start the first session of today's conference with our first speaker, Professor Dr. Kumar. I think Dr. Raj is also connected to us. Sir, are you there? Dr. Raj?
Minute. Just a second, right?
Okay, sir. Dr. Raj is an esteemed speaker of the conference. He holds a PhD and MS in pharmaceutical sciences with specialization in medicinal chemistry from Naipur Mohali. He is a distinguished professor at Central University of Punjab, Bhatinda, with an extensive background in medicinal chemistry and drug discovery. Dr. Raj has made significant contributions to anti-cancer research, green chemistry, and the development of diagnostic agents. With over 108 publications, 6 patents, including 2 US patents, 7 book chapters, and 3 books to his name, Dr. Raj Kumar's research primarily focuses on targeting EGFR, HDAC, and PKM2 for cancer therapy. He is also exploring innovative approaches such as antibody drug conjugates and PROTECT research. His excellence in research has been recognized with prestigious awards, including the 8th Visitor Award in the year 2023 from the President of India, Srimati Tripathi Murmu, and the Shivnath Kohli Best Researcher Award in the year 2024. Dr. Raj has supervised numerous PhD and MPharm scholars and has secured research fundings of approximately 3 crores from different organizations such as DST, SERB, UGC, and several industrial collaborations. As a fellow of Royal Society of Chemistry UK, he actively reviews for high impact journals and grant agencies worldwide. His expertise and dedications make him a leading voice in pharmaceutical sciences and an esteemed speaker at this conference. Please join me in welcoming Dr. Raj Kumar. Sir, will you please present share your screen for the presentation purpose?
Thank you very much.
Okay,
Can you hear me?
Yes, sir. Yes, sir. You are audible.
Yeah. So I would like to first thank to organizers of Rohru College that they have invited me at this event, and I'm sorry that I have joined virtually, and I am at home because today is holiday, but I am working for a thesis right now. So anyways, uh, thank you so much, and I'm happy to hear that it is first international conference at the government college. I have few students also from your college, and I have taught them, and you people are really doing great. So and I saw that there are more than 400 participants have joined. That's a really good achievement. So I congratulate all the organizers of Government College Rohru for organizing such a wonderful event virtually, as the work is also virtual. So let me share my slides. I'm just new to this WebEx. I'm just checking it. Oh, wait a minute. Entire screen, I guess I can share. Or window. I'm not able to share this thing. Can you help me in this? Like, I have joined as a guest. So can I present?
Yes, sir. You can present, sir. I have
So give me the access so that I can present. Otherwise,
already. I have given already access, sir. You are presenter as a presenter.
okay, then PP Chrome tab. It's not showing PPT, but it is showing the Word file.
Share screen. Share as a screen.
Yeah, entire screen is not doable here. It's not, the icon is, yeah, now I can do that. So yeah, can you see my slide?
Yes, sir. Yeah.
Yes, sir.
Okay.
Visible, sir.
Yeah, now it's visible, right? The full show is also, full slideshow is also visible, right?
Yes, sir.
Okay then, dear participants, first of all, I would like to say that I am not a computational chemist. I am a medicinal organic chemist. So this is my disclaimer. But yes, I use in silico tools for finding out the fundamental things, that how drug is binding, pharmacokinetics and dynamics aspects. So one must, as a medicinal chemist, everyone must know all these tools, for their help in drug discovery and development. As I have mentioned the title, in silico drug design and case studies, I will be touching upon some of the fundamental aspects of the drug discovery. So what do you understand by drug discovery process? If you talk about drug discovery, is such a complex process. It is not a single process, and it is not driven by only chemist or biologist. It is combination of all this. It requires actually multidisciplinary and transdisciplinary aspects of knowledge sharing and literature, and coming out with all the things together on one platform. So if a drug is discovered, it is not the only forced through a chemist or a biologist or pharmacist. No, And pharmacy itself is a interdisciplinary science, of course. So if you see the drug discovery aspects, you will see that you need to know the target. Who discovered these targets? So biologists, chemists, together they discover the targets. And you need to know some of the targets, like there are many targets and mostly they are proteins, steroidal, other targets, but protein targets are there. And then if you know the targets, it's not sufficient. You know the structure of the targets. That will further help you to finding out, the things. But in a classical drug discovery process, we know the targets, then we design something, some compounds hypothetically, or just hit and trial. We synthesize thousands of compounds, and then we screen them, and we found out, okay, there are some hits, we optimize them and then convert them to leads and further lead optimizations for particularly pharmacokinetics parameters like ADME, and then it goes to the animals and then goes to the further clinical trials. This is a typical drug discovery process for a molecules. Like if you start with 1000, you may end up with 2 or 3. Even then they may fail into the phase 1 or phase 2 or phase 3 clinical trials. And this process requires long time, of course, and a lot of money, billions of dollars, for this drug discovery process. And can we reduce this time, like from 12 to 15 years, to shorten it to, let's say, less than 10 years? Can we do that? Yes, efforts are being made in recent past to reduce this process, not only in this target discovery, but at every stage of drug discovery, we can utilize, at least I'm sure up to preclinical stage, we can utilize this virtual screening aids or in silico tools, and now people call AI or other, machine learning programs, to reduce this time at each stage, whether it is pharmacokinetics or dynamics. Okay, so let's the people earlier call it CAD, the classical CAD, computer-aided drug design, can be utilized in any of this, phase 1 or phase 2 or preclinical. This is all preclinical stages. What do you think that for a computational aided drug designer, what is the requirement? Like what one should be? So one should be medicinal chemist or biologist, or I'm sorry, somebody is talking behind. Okay. Okay.
All the participants are requested to mute yourself.
Okay. So you see that genomics, pharmacogenomics, and genetics play a very important role. So if some genes are there, they are known, and mutations are known, we can think of, and gene products like proteins, if we know the structure of proteins, here bioinformatics comes into picture. That is also a part of CAD. So various, you should know the primary, secondary, tertiary, and quaternary structure of the proteins. And you should know how many amino acids are there, and where are the binding sites, and how many, what are the cofactors there, FAD, NAD, and some metals present in the proteins. I know many of the students enter the lab and they just want to do this fancy thing, CAD and all this thing. They just want to publish with CAD, but they do not know what is a protein and where is the catalyst site and where is the other allosteric site, and what are the amino acids. Even they do not know the structure of amino acids. So without this, you cannot think of, like, it is just hitting around the bush and just getting something fancy. And no, you should know the knowledge of informatics tools. The entire structure of protein is important to know. Okay, then you have target validation. So you have a lot of targets, okay, but you need to know the targets which is actually responsible for the pharmacodynamic action and which is responsible for the disease. For example, people work on kinases, or there are more than 500 kinases, but many of them are druggable, some are not. Okay, so target druggability is required in case of the CAD techniques. Then you can have a, you can design your own compounds hypothetically, or you can take help of, some available databases like Sync databases or PubMed, PubChem, whatever. You can design your own library of the compounds. If you are a chemist, you know that, okay, this is the pharmacophore. We can build up the, structures, and we can enumerate it, and we can design a number of compounds, more than 100, 200, 5,000, whatever. Because it is a virtual thing, you can think of as much as possible, provided you should know the synthetic feasibility of the lead compound. Otherwise, everything will go waste. People come up with some, some things, but they are not synthesizable. Sometimes retro is not possible, and sometimes, starting materials are not available, and some techniques are not available, or some methodology is not available. So we have to think logically in that way also. Then you have you can assess them by some procedure and dock them where is the active site, and then find out where you can do it, and find out some score based on energy or something. Then you can easily make them available at the protein, and find out how much binding it is. If the protein structure is known, you can make the de novo design also. You can think of the pharmacophore mapping techniques also, if you know the pharmacophore, and you can get some target feasibility also. The another important thing is that in lead optimization, you can make use of QSAR or 3D QSAR or structure based other techniques also. Okay, and in the preclinical testing also, you can predict before the preclinical actually starts. You can predict their ADME, toxicity, cardiotoxicity, and absorption, distribution, metabolism, like metabolic stability is required with respect to the drug when the drug goes into the plasma and it passes through the first pass metabolism. How much it is inhibiting with the cytochromes P450s. There are various variants of the cytochrome P450s, and the renal clearance and other things is required. Now many softwares are coming up with the protein binding, plasma binding, other things. Then physiological based pharmacokinetics simulations we can do. So in this time, this CAD can help from, it's like a synthetic chemist. With all these softwares availability, we can minimize the waste, we can minimize the number of compounds. So of course the economy, the compounds will be economical, and like we can reduce the number of steps. And this because in all the steps, if we can find out a compound, if we know one tool where we can apply all these parameters and get some good lead. Of course, it has to be tested in vitro, then only proof of concept can be proven. And so a drug candidate, either way, the typical drug design approach, like create a new molecule, dock it, analyze it, and then estimate the binding strength, and then estimate drug-like properties. Then it can be or may be a drug candidate, may or may not be. Okay. So this is a fast, high throughput screening approach, which is called virtual screening. And it's a computational techniques. We can design number of compounds. So this is the protein, it's a macromolecule, and it has some site, which is a cavity, and where a number of molecules you can hit them and find out, let's say 10,000 compounds, and which has drug-like or lead-like properties from the databases. And then it will narrow down as a funnel shape. You can see that narrow down and narrow down and narrow down. And like nowadays, pains is coming into picture. If you read the JMedChem paper, JMedChem does not accept those compounds like features, like those compounds which have activity in all the assays, but in actuality, in vivo, it fails. So that filter also you can apply in softwares. And then ADME properties also you can like it should have a ADME properties, good bioavailability. But I have seen some softwares calculating 100% bioavailability of those molecules also, which are actually not bioavailable. So you have to be smart enough to not rely simply on the softwares. you have to use other softwares to compare, and because all softwares work on some algorithm, so you cannot just blindly, you know, just trust them. So you apply your brain, and then the sum of the compounds, top scoring compounds, maybe top 10 or top 15, you analyze them, see them, and find out their synthetic feasibilities, or if they are available, buy them and test them with respect to this particular target protein in vitro, followed by cell-based and other things. And if the logic comes out to be correct, the order is as per the in silico results, then you are lucky. But in vivo, in cell based, like chances are less, because the other things are also playing to play important role. And if we go by, there are drug design approaches, structure based drug design or ligand based drug design. This is the typical flow chart where how we use the, you know, structure based. The softwares can do many things, but you have to be smart enough. See the active site, see the protein, and dock your compound. See, this is a molecular target, do the molecular modeling and this thing. Is it very simple? No, you have to first prepare. This looks like very simple, but you have to find out in the background so many things. Two things, because you have to dock, like you have to take protein as well as the ligand. So you have to prepare the protein as well as the ligand. So protein, you have to prepare the protein and you have to prepare the ligand so that they can bind and minimize. And you have to, the result which come out as a docking result is a energy minimized structure. Okay, and you will see that various interactions with this thing. This is a structure based approach. We'll talk about MD simulation also. Then you have, if you do not know the structure of protein or you want to optimize the lead, you can make use of ligand based approach, where you have the ligands, you optimize, you make them, divide them into two categories. One is, you know, training set and test set, train them and put some descriptors, and you can generate a model like QSAR 2D or 3D QSAR model. And then you can find out, okay, which is favorable region, which is not favorable region, and you can identify the leads and synthesize some new compound based on that QSAR model. Or you can go for scaffold hopping, or, you know, ligand based pharmacophore. You can make like ADDR or DDDHR, like different pharmacophores you can pharmacophore approach you can use. Okay, so it is very important that you choose a target, then determine 3D structure, analyze binding site, and dock the compounds through a database or whatever you have synthesized some compounds. And then the compounds which come through the filters, test them under in vitro conditions. So if it's a lead compound in micro or whatever you're looking for is fine. If it is not, then you can further modify the lead and then again redock it. Okay. If the structure of the protein is not available, if there is some similar protein is available, you can develop a homology model also. Yeah, like where there is no proteins available, either you can go for homology model, or the based on leads you can go for the QSAR approach. And if it is a lead molecule, you can determine the 3D structure protein complex. You take the compound because you have tested it, allow the biophysicist or X-ray crystallographer to get a beautiful co-crystal structure and see how it is binding. That will serve as another protein complex which can be used by the other people to screen them. So then based on that, you can further design it or further get a nanomolar inhibitor. So that would act as a potential drug candidate, and which has to be tested for further ADME studies to become a drug compound. So this is the way of, you know, people think of in a drug discovery process. So in molecular docking, what do you need in molecular docking? You need to have software, of course. You need to have two things. One is protein, other is the ligand. If you are multiple ligands, ligand means a drug molecule or a small molecule which you want to bind with the drug. And if that molecules, there are more than one molecule, 2, 3, 4, 5, 6 at a time you want to give, that becomes a part of kind of virtual screening. So docking is a computational techniques. So where the binding, and so after docking, you will arrange them or see its interaction and compare with some standard inhibitor. Of course, you have to put a positive control, which could be a inhibitor or agonist or antagonist based on the case of the molecule. And you see that a small molecule, how a small molecule is bound in the protein. So it can have a number of orientations, conformations. So because a ligand, you know the structure of a, if you are a chemist, you know the structure that a small molecule can have a pi bonds, can have a sigma bonds, C double bond O, NH. So there are some rotatable bonds. So molecule can enter into the cavity and it can sit in a different ways. You can see that this blue one is a standard ligand, and the other are the design compounds which are occupying the cavity where standard positive control is there. So you must know. So as you reach the cavity, so that means in the analysis you should put focus on the cavity, how your molecule is sitting in the cavity and how long it will stay in the cavity. There are more important points to understand. So how it is sitting, that means the energy should be minimum, that complex is favorable, and the thermodynamic chemist, they find out the delta G, that how much is the minimum energy to which it is binding. And this binding is a result of many forces. That means a drug is sitting into the cavity not by itself. It has many interactions or forces possible with what? With the cavity. So what is this cavity lined by? This cavity is lined by various amino acids. So that means you should know the structures and some side chains of the amino acids with which they can interact with our drug molecule, and how we can think of modifying your small molecules by just changing some of the functional groups or putting some linker to make more binding possible with the amino acids. So that means you must understand the structure of protein. What is the primary structure of protein? So it could be in a polypeptide chain, it could be a nonapeptide, decapeptides, so they can act as receptors. So that means you should have to have knowledge of biology as well as chemistry. That will, foundation is important, fundamental knowledge is important to start with. So primary structure, secondary structure, and then quaternary structure, like hemoglobin has this quaternary structure, and tertiary structure. Some proteins are active in tertiary structure, some are active in quaternary structure, some act as a nonapeptide, or like 51 amino acid. Insulin is also available. So you should know the structure, and you should know the cavity or the binding site. I would say the binding site, of the protein also. So as I mentioned, that these are all proteins is made up of amino acids, and these are some of the amino acids, like 20 natural and then sometimes unnatural amino acids also people synthesize and then incorporate. So you should know their structures, their side chains. They're like methyl, or some, like it is serine has side chain CH2OH, and threonine has hydrophobic methyl as well as the hydrophilic OH. Then cysteine is very important amino acids, and then valine, leucine, isoleucine. So you can categorize them based on acidic basic amino acid, based on hydrophobic hydrophilic amino acid. So based on the side chain, you can think of this thing like basic amino acid like arginine, lysine, and you can see the epsilon NH2, which is protonated at the physiological pH. And then arginine has a guanidine, and then histidine is having also, this thing. And then tryptophan has an indole ring. Tyrosine, this is very, very important amino acid in case of the kinases, where tyrosine plays a very important role in carrying out this taking away the phosphate group from the ATP, and it carries phosphate. So phosphorylation takes place at this oxygen, and then which is transferred to the other proteins in signaling pathways. And if you see this is tyrosine, so there are tyrosine kinases, there are serine kinases also, because this is alcoholic OH. Of course it is alcoholic, less acidic, but it also catalyzes phosphorylations things. So if serine is present in the cavity, of the ATP binding site. So serine can also accept the phosphate group. And serine kinases, threonine kinases are also, there. And then threonine also has OH side chain, if you can see both. And cysteine and like guanidine, sorry, lysine and arginine, these are basically more abundant in case of some of the proteins like chromatin protein, okay, which wraps around which the DNA is wrapped, like histone proteins. So you should know that each protein is unique and the ratio of all these amino acids vary in many of the proteins. Some are rich in histidine, some are rich in lysine, some are rich in arginine, some are rich in. So there is a purpose of this richness because they have a protonation at the end. Some are having rich in CO negative, some are having asparagine, glutamine. So these are important. So as I mentioned that in kinases, you will see abundantly serine, tyrosine and threonine kinases, which cover more than 900 proteins structure like serine kinases, threonine kinases, CDKs, EGFR, or other kinases, okay, VGFR, EGFR, etc. So that means stereochemistry as well as the side chain functionality and their nature, you must know that. We will see that in the cavity. Why I'm teaching or why I'm telling all these things, because if one of the amino acid is present, let's say this amino acid is present, or let's say tyrosine is present. If tyrosine is and it is very important for the activity, or let's say threonine is present and threonine is replaced by methionine, and you know that methionine is more bulkier also, it has a side chain, one more carbon and s methyl, and this is, if this is replaced with this, so earlier the drug which was binding with this OH and having hydrogen bond acceptor with making hydrogen bond with this H, now it cannot make. So think about if you have an amino acid which is important in the cavity, and it is mutated or replaced site, this is called site mutation, is mutated with another amino acid, will drug be able to bind? No, because drug, sometimes these amino acids are present at the gate. So if the gatekeeper residue is changed with another amino acid, then the scenario will change. The drug may be less active or more active. But if it is replaced, in most of the cases, it is less active because it is a method, or it is a mechanism of the cell to induce resistance to a particular drug. So see the structure of these amino acids, and you should be thorough by heart their structures so that you can think of designing a drugs and thinking of interaction with them. So you can think of that like valine, alanine or something, they will induce having hydrophobic or van der Waals interaction. Phenyl, if they are having phenyl or indole kind of thing, they may have a pi stacking interactions with the drug molecule. The drug aromatic ring can overlay with the benzene of these rings and have a pi stacking interactions. Or if our drug is having some salt or something like that cation, and it can have arene cation interactions because the pi electrons can be donated to so it's a kind of complex thing, metal complex. And if there is a hydrogen bonding donor or acceptor like OH, it could be accept, it could be donate. So these side chains are really, really important to keep the drug engaged in the binding site. Imidazole also plays a very important role. And based on this histidine, the cimetidine and ranitidine discovery, breakthrough happen. So these are the each amino acid has its history and, important things to understand. Once you know the amino acids, then how will you get the structure of the proteins where you want to dock? So what do? You think that software brings their structures? No. It's good to have a X-ray or cryo-electron structures, the 3D structures available from the protein data bank or other data bank. So these protein data bank, they're actual structures, but sometimes what happens, these structures are not minimized properly, or some of the bonds are also missing. Some of the things are also missing. So that will help, that you have to take care while preparing the protein. So you get a protein complex, and then this is the protein structure which comes out through the X-ray crystallography. This is a 3D structure. It may be or may not be having a ligand. It could be a endogenous ligand, okay, like acetylcholine inside the thing, and like AMNP inside the kinase. So it could be endogenous ligand or a inhibitor, whatever the case may be. But but both of them will help you to design new compounds. Like in case of xanthine oxidase protein, you can have a complex structure of allopurinol with xanthine oxidase protein, and then you can see the structure and dock. You can put your own compounds there and see the interactions. And if you get better interaction as compared to the allopurinol, then it might be a good compound to start with. So NMR also is helping fragment-based drug design and other things. NMR spectroscopy has also proven very nicely for target structure identification in this case. This is one of the proteins which we have recently published in JMED Cam. Like this PKM2, it's a protein kinase, but it is a metabolic kinase. It is not a receptor kinase, and it is also involved in cancer, carcinogenesis. Overexpression involves, and it can exist in a dimer or tetramer, both the forms. So this is a dimer of a protein where you can see that there is a kind of C2 symmetry. And if you see, it has a various binding domain. One is fructose binding domain, where fructose can bind and activate it. And another active site is there where you can put some inhibitor or to inhibit its activation. And these are the important amino acids which are lining, this thing. And you can see that there is at the two dimers where the monomers meet at the dimer junction, there is another activator binding site which one can explore, and these are the amino acids available. So before starting docking, what you have to do, you have to first download the protein, see its structure, read the paper with respect to the protein, because generally these papers are published in Nature or Biochemistry, JBC, or other good journals, where then you have to take that protein which has a good resolution also, because there will be many complexes or many proteins available. Get along with the proteins, see all the amino acids, see the active site. Just go through before starting any docking study. Just visualize the proteins in 3D and see all the amino helices, ribbon shapes, things of the protein. Then see where we can,
400
where are
meters,
the
turn
bindings,
right onto Vanasthali Marg.
and how the drug is, bound to it. Okay, so that is really important. Then if you think that some of the protein, bonds are missing, just prepare the protein, minimize the energy. Then on side by side, you have to prepare the ligand also. Your own, either it could be one ligand or multiple ligands, and then you have to choose them. So that means you have to download a protein from as a PDB file, and then you have to see anything is there or not, and you have to prepare the protein. This is the various methods to prepare the protein, and then you have to start the docking. But before starting the docking, you have to first find out the RMSD as well, like whatever the inhibitor or the agonist present inside the protein should also go back. You remove it, draw it or remove it, and again dock it. And if you get RMSD very good value, like less than 1 or something, then it is very good. Less than 2 is also considered good. That means it is overlapping with the same thing. So your protocol, your model, or your strategy, your methodology is perfect. If it is not, then try again, and you have to generate a grid and find out. So these are all software based things. Logically, you have to think, you have to see the proteins, you have to take a tour of the protein, and then dock the compound. And then after this, you have to analyze the results. Results are generally I ask students say, it is showing very good score, but you should know what is that score, how the protein is, how various amino acids are interacting with the drug molecule. So sometime I have seen good score also not giving good alignment or good binding also, because you have to visualize each and every pose, and you have to see various conformation, because in ligand preparation, you will see the compound will have a different various conformations, and software chooses the best conformation to dock. So this is the thing. So this is in molecular docking, you will see a various number of methods like rigid docking, flexible docking, induced fit docking. So there are softwares available. or each one is having pros and cons over others. So now people mostly uses induced fit, dockings. And in flexible or in rigid docking, people think that okay, protein is in a rigid form or this thing, but everything is in dynamics. So you know that in biology system, proteins is not sitting in one place, as so is the case with ligand. So induced fit docking is mostly used. I'm not talking about the software detail. I'm just talking about. So here also it is example of my my work, my lab's work, which we published in journal. So this is you can see that the red color is, you have to see the alignment in docking also. See, the red color is erlotinib and the green color is my drug. So it should have at least alignment that it shows, this figure A shows that our designed compound is occupying the same cavity where already erlotinib is present. And second figure shows the interactions. So as I told you that earlier also, that EGFR also exists as having is a wild type and mutant type. What is the difference? In wild type, threonine is there, and in threonine 790, but in mutant type, methionine is there. But if your drug is able to interact with both, that means it is good. So you can see that this drug, this compound is able to interact with methionine 790 also, methionine 790, so which is a gatekeeper residue. There are many possibles, but you will see that hydrogen bonding, methoxy is showing hydrogen, but they're weak hydrogen bondings because you need to have a strong electron withdrawing group or fluorine or something like that. So various forces you must know as a chemist. What is hydrogen bonding? What is the distance? Hydrophobic interaction, pi-pi interaction, metal coordination, pi-cation, all kinds of interaction you must know. You may not be having all of them in one figure. It's okay, because in some of the metalloenzymes like xanthine oxidase has molybdenum, HDAC has zinc in its cavity site. So if you have to carefully see whether your drug is interacting with the zinc or with the molybdenum already present in the site, and is it disturbing the geometry of the metal which is already present in this thing. So you have to carefully see each and everything. Just do not come out with the result that, oh, I have got the good docking score. That may be wrong. So you have to see that your compound might have docked somewhere else, or it is not having. So you have to be very, very careful, very cautious when you are analyzing the computational results. Okay. And this is another figure which is example of because most of the kinases are beautiful to explore for computational drug design, because they have many things to talk about. So they have DFG motif. DFG is nothing but aspartic acid, phenylalanine, and glycine. So the three chains are available, and if you see that it has many sites of binding. So in EGFR, it's a simple kinase, not EGFR, and just any kinase. Any kinase can have one ATP binding site. This is ATP. If you replace your ATP with if red color is your drug, so it is occupying the ATP binding site. And you see that there are side chain, which is an activation loop. This is a DFG loop, where DFG is attached to the while binding. It is coming out. It's a part of the loop. So it is DFG active. So that inhibitor would be called as type 1. But when there is no interaction, so it is with respect to the active conformation of the protein, it is inactive conformation of the protein. So in inactive, even if your compound is able to occupy the ATP binding site, even if it is inactive or ATP DFG out, it will be called as a type 2 inhibitor. See how the classification of various types of inhibitor has been given in the same protein, just based on the binding and the DFG active loop. And if it is binding any of these sites here or there, then which is not binding to the active ATP site, then it is called allosteric site. But this allosteric site is within the ATP binding site because it's close to this. Because the whole is the ATP binding site, this one, and this is near to the, so this allosteric site is near to the ATP. But this allosteric site is away from the ATP binding site. So this is another type 4 allosteric binding inhibitor. And if you can design a drug which is a very big molecule and or same or a one compound, single dual compound, so it can occupy both the ATP binding sites as well as the other allosteric site, it will be bivalent, type 4, type 5. And if it binds irreversibly, that means think about now how it can bind irreversibly. That means your drug is having some functional group and making a covalent bond with one of the amino acids lying in the cavity here or here. So now we recall the various structures of the that's why the structures of amino acids is important. So that covalent interaction is generally with the cysteine, which is having its SH, or it can use OH or NH, but in a rarely case, but SH is very predominant in making covalent interaction with your drug molecule. So you can, in this slide, the moral of the story is that your drug can be type 1, type 2, type 3 or type 5, 6, whatever, based on the binding. So you can classify your drug. So that means so many options are available. So that means this is a fundamental that whenever you are looking any protein, you should first see where are the binding pockets and what are the reported cases with respect to that protein. Then you should start your work. And this our drug so this is our slide where we have shown that we can have a various PKM2 inhibitors occupying, and you can see that the magnesium is there, which is here, there, and near. So magnesium can also coordinate with the phenyl ring. Remember I talked about arene cation interactions. So if our drug has a aromatic ring, it could be naphthyl, phenyl, pyridine, whatever, and it can make an interaction with magnesium as well, or molybdenum well, or whatever. So this is also important. So if we talk about molecular docking softwares, all of you might be knowing it. There are many softwares, freewares, and paid softwares are also available. This is the list you can go through. And you can go for a QSAR technique also, quantitative structure activity relationship. This activity could be anything. It could be any property. It could be toxicity. It could be, it will become QSTR. It could be any even pesticidal activity is also possible. Now I have seen many of my friends who worked in computational things. They are working for a pesticide company where they want the toxicity. Here we do not want the toxicity, but idea is same. They are generating a contour, and in those contours they are finding out which is favorable region, which is not favorable region. So in QSAR or QSPR or QSTR, you need to have a set of compounds which are already having activity, and they should have a perfect alignment, and then basic toxicophore or pharmacophore should be same, and then here their changing must be there. Second important thing, the reliability of data, because AI and machine learning will be unsuccessful or fail in those conditions where the reported data is not reliable. If you are taking any JMedChem paper or good paper where IC50 value has been given, and you are using that IC50 value as a tool to train the next compounds, and if those IC50 values are falsified or there are problems in the biological data, then your results will not be good. So you have to be cautious in that way. So you can generate, and another approach is pharmacophore modeling. If you know, like classically people used to talk about acetylcholinesterase inhibitors or estradiol receptor inhibitors, like estradiol length of the one phenol to another phenol, and then they want to check. So here you can do it computationally also, that 3 or 4, like this is 1, 2, 3, 4, 1 aromatic, 2 aromatic, hydrogen bond donor, acceptor, or aromatic. So like that you can design a pharmacophore based on the structure of the ligand, not the protein, structure of the ligand. You can calculate the distance, and based on the distance you can design a new pharmacophore, and then do the search also for new compounds which fit into that, and if that is the affecting property. Okay, and here people replace, like it is a ring. If this ring can be replaced by naphthalene, phenyl, here it is not defined, but you have to define. This ring could be bigger size ring, acridine. What is the size of the ring required? That is also important. Then if you're writing hydrogen bond donor or acceptor, if in your main compound OH was there, can it be replaced with SH or NH2? So that you have to think very carefully. Okay. Now people are talking about artificial intelligence. This paper came in 20, I guess. This team, Strubble and this team, they synthesize, they worked on it, this current future roles of, and they published a perspective. One can go and find out what is the usefulness of this AI. Then artificial intelligence and machine learning in drug discovery and development. So novel approaches are also coming. People are calling neural network. So application of artificial in drug discovery, they are coming up. AI in drug design, prediction of, so you can predict the 3D structure of target protein. You can predict drug protein interactions, de novo drug design, physicochemical properties, bioactivity, and toxicity. So it is actually a assimilation of CAD, and then it's further whatever data is being published. I know some databases where somebody wants to work on like COX-1 or COX-2. So in discovery stage reported compounds and their IC50 values, actual IC50 values are deposited in the database, and then one can go and download that library and train the molecule and may use it for some other purpose like repurposing, or for virtual screening, or for developing a new and apply the AI methods and find out a new lead, because the data is already there. Now I'm again saying the AI will never work if the data is false. So the results of AI will not be good if the deposited data is not good. So whatever you give in, it will come out. So it is a GIGO rule, garbage in, garbage out. If the things are not good, it will be garbage of no use to you. So machine learning models you can develop, and selection functions, orthogonal computation. So you can train them and find out a good leads. Provided the data feed or the thousands of molecules are there where actual IC50 is there, then it makes sense, and you may end up, through the AI methods, you may end up with a good lead molecule. So I told you that in silico methods are applicable now everywhere. Even I have seen in drug metabolism, in silico prediction of cytochrome P450 approaches and protein data bank. So these are the, it's everywhere you can see that. I tell you, like some of the, in drug resistance, I presented one case of drug resistance where one of the amino acid was mutated, right? Another could be like PGP, protein, this P-glycoprotein efflux mechanism. That also works by, PGP also works by this, sometime mutation or some, it kicks out the molecule because of a flexible binding site. And if the drug doesn't permeate, there are some transporters. Transporters are also made up of, some of the transporters are also made up of proteins. So if that some becomes substrate of the transporter, sometimes transports the drug binds to a transporter, but it doesn't go inside this thing. So that docking interactions you can also see through, and you can propose that the penetration or permeability of a drug based on transporters. So these are some of the tools in drug design, which are, this is a huge list. I can provide you after this conference also to the students or to the participants. So these are the chemical databases which are available. And I have one more thing to share with you. I have a Word file. Yes, this Word file, this I wanted to paste it in the PPT, but so this is a databases. It is 75 pages list where my group has assimilated all the information of the available databases, freewares or commercially paid, this thing in one go. So the softwares for protein ligand complexes, and then we have softwares for, I can share it with you, target databases, pathway databases, many databases are there which people who are working actually in AI or some other, they can make use of it. So in ready-made one place thing you can get. Second, nowadays people are working on protein degraders, which are a very big molecule. I have started also working. There are also some softwares are coming up where there is a ternary complex, 2 proteins and 1 big molecule. They stay together, and some complex structures are also reported in X-ray structures have also been reported. So this is a big list of all the tools in drug design. I can share it with you after this conference, or yeah. So this is a big list. So thank you very much. Thank you so much. And there are many techniques I cannot cover. It was just a fundamental thing that the thumb rule is that you have to understand the chemistry knowledge, basic biology knowledge, if you really want to work in computational field. Otherwise, it will be just wishing, what do you call, revolving around and there, here and there. You will not get any concrete things in hand. Thank you very much.
Thank you, sir, for delivering such an insightful and engaging session on the basics of CAD. Your expertise and valuable insights have truly enriched our understanding of the topic. Now, we will invite questions and discussions from audience. Anyone want to ask any questions? Any participants who want to ask any questions?
Hello.
Yes.
Yes, ma'am. I have one question.
Okay, you can ask.
Uh, Hello. Good morning, sir.
Yeah, good morning. Good morning. I'm sorry, I was reading
Yes.
the comments. Okay, yeah.
Sir, I want to know about RMSD value.
Yeah, it is root mean square deviation. So what you do, you extract the ligand which is already present in the protein and redock it, and see in the pose you will see that uh, there is already drug, and the same drug you have docked it. If it is binding like overlay structure is kind of this thing, perfect, then there is less deviation. Okay, if there is less deviation, that means your method of docking is all right. We can use it for your now own ligand molecules. So this is a kind of, so that is a kind of proving or proof for the thing. Are you getting my point?
Okay, sir. Sir, how we can write in paper what value we have to select?
In paper, see, if you use the software, it will give the value and you have to write the, this is called a validation protocol, validation of the molecular docking. This is a procedure called validation of the molecular docking. You can find out many videos on YouTube that how to do it. So just do it and then the value will come. RMSD value will come, you have to report it. Okay, that's it, that you have done the validation.
Sir, if RMSD value is 0, what it indicates?
If it is 0, it cannot be 0. It will tell that if it is 0, that means there is no deviation. It will be very much perfect.
Okay, sir.
Right?
So, sir,
It is
so
not
we can
feasible
prefer
actually.
0.
Hmm?
Sir, can we prefer 0 value?
See, preference is different, but it will never come. It will never come, but it should be less than 2 or 1 people. Sometimes it is very difficult to achieve less than 1 also. Okay, RMSD. So that is minimum deviation from the already reported positive. Both are positive control, same thing, same drug. For example, I give you example. You have a xanthine oxidase protein, which you know it is being responsible, overexpression is responsible for gout. Okay, so xanthine oxidase protein, we download a protein where allopurinol is already there. So you remove the allopurinol, again redock it. It's a virtual thing. So if the already present allopurinol and your allopurinol is overlapping exactly, okay, still the value will not be 0, but will be close to 1 or something. You can use it. Use this method. Same method you have to apply for your own ligands which you have designed yourself. Okay.
Okay, sir. Sir,
Okay.
as we see in many paper, there are RMSD value like 1 point something, 1, 2, 3.
Yeah. That is acceptable. I'm telling you, I think below 2 is also acceptable. No problem. But it should not be that 3, 4, 5. No, that means there is and you will see that if it is 6, 7, then the molecule will not sit overall. It will be, away. That's why you have to apply your mind. You have to see, visualize it.
Okay, sir. It means, okay, sir. Okay.
Okay.
Distance.
Distance, yes, also, and the alignment. That is more important.
There is one more question in chat.
Hanji?
Kindly ask one by one.
मैं आपको एक और बताता हूँ बात. See, YouTube में जितने भी beginners हैं, any beginner, please see there are many good lectures available on YouTube. Okay. मुझे कुछ समझ नहीं आ रहा. Okay.
उधर नहीं थी, इधर करवाए. Disturbances. Only simply ask your question if you have. No more questions?
मैम प्रो ड्रग डिजाइन के बारे में सबसे जानना चाहते हैं.
First introduce yourself and then ask the question.
Ma'am, myself Utkarsh Kondan, representing Government Pharmacy College Kangra, and my question to sir is about the pro drug design.
Pro drug designing.
Hello.
Yes, ma'am. Good morning, sir.
हाँ, good morning.
Sir, I have a question regarding pro drug design.
Sorry.
Give a brief. Sir, can you give a brief about the pro drug design?
We want to ask about pro drug designing.
Which
Yes,
drug? I'm sorry.
Pro drug, sir.
Pro drugs.
Pro drugs.
Okay, pro drugs. Okay, yes. What do you want to ask?
Sir, how different softwares help us in designing the product, and we have.
I'm writing a perspective. I'm writing a perspective for product designing and want to publish in Nature Protocols. I could not find softwares which help in designing a pro drugs. So you have to apply your, medicinal chemistry knowledge for that. Pro drug.
Is there any software that will help us in pro drug design or not?
That's what I'm saying. I'm saying, I'm telling you the answer. I didn't find any software to design pro drug.
Okay, sir.
Because pro drug, see, I'm sure that some software will come into market in future, because in pro drug it requires where you want to deliver the things. In pro drug also many aspects are there. Some like ester hydrolysis, reduction. Because pro drug uses the environment of the cells, right?
Yes,
Acidity.
Sir, can you tell about the monoclonal antibodies also? Can we design a monoclonal antibody?
नहीं, monoclonal antibodies नहीं, ADCs हैं, antibody drug conjugates. Monoclonal antibodies के लिए डिजाइनिंग के लिए नहीं है हमारे सॉफ्टवेयर.
सर तो
जो
ये
ADCs हैं,
ये जो ड्रग कंजुगेट्स आप बता रहे हैं हमें, ये मतलब हमारे.
मैंने अभी कोई भी ड्रग कंजुगेट्स नहीं बताया आपको इस टॉक में. मैंने आपको बताया था. हाँ, बोलिए.
आप मतलब कि ड्रग कंजुगेट, मैंने जब आपसे एंटीबॉडी का पूछा तो आप मतलब कि ड्रग कंजुगेट्स बनाते हैं. तो सर कोई सक्सेसफुल ड्रग कंजुगेट जो डेवलप किया गया?
नहीं, ड्रग कंजुगेट बनाते हैं. एडीसीज तो बहुत हैं. देयर आर कितने अप्रूव्ड हुए हैं मार्केट में. देयर आर ड्रग कंजुगेट्स, एंटीबॉडी ड्रग कंजुगेट्स. अभी पिछले 2025 का मेरा पेपर उठाओ जेमेड कैम में, देयर आर मोर देन 10 एडीसीज आर एफडीए अप्रूव्ड कैंसर के लिए पर्टिकुलरली.
यस सर, आई वांट टू नो फॉर द कैंसर पर्टिकुलरली.
हाँ.
यस सर, मतलब कोई एंटीबॉडी ड्रग कॉम्प्लेक्स या एंटीजन ड्रग कॉम्प्लेक्स जो कैंसर में जिसका जो क्योर रेट है वो काफी सक्सेसफुल है, कोई ऐसा हो जिसका आप एग्जांपल बता सको?
नहीं, एडीसीज का सक्सेसफुल थोड़ा ज्यादा है क्योंकि ये दो तरीके से काम करता है. ठीक है. अब मुझे एडीसीज का जैसे टेस्टोमुजाब वगैरह वो तो मोनोक्लोनल एंटीबॉडीज हैं, लेकिन उनके साथ अगर हम एंटीबॉडी ड्रग के साथ ड्रग भी साथ जोड़ दें तो वन एंटीबॉडी कैन डिलीवर सो मेनी ड्रग्स. कैसे? बिकॉज एंटीबॉडी इज ए प्रोटीन, बिग मॉलिक्यूल, और उसके ऊपर आप कंजुगेशन लगा के आप मैनी ड्रग मॉलिक्यूल्स जोड़ सकते हैं. ठीक है. तो उसमें टेस्टोमुजाब या कोई और अगर हम उसके ऊपर बहुत सारी ड्रग्स लगा सकें तो एडीसीज के तो उसमें एक्टिविटी कैसे बढ़ेगी. आई टेल यू. वन, जैसे काइनेज है. काइनेज में जो बिकॉज इट्स द रिसेप्टर काइनेज, जो एक्स्ट्रा सेलुलर है, देयर मोनोक्लोनल एंटीबॉडी विल बाइंड एंड इंट्रासेलुलर में ड्रग विल बी डिलीवर्ड, एंड इंट्रासेलुलर में एटीपी बाइंडिंग डोमेन है. सो इन वन वे टू थिंग्स विल बी, एंड इससे रेजिस्टेंस कम हो जाएगा. So that could be that is the approach ADCs. और इसमें ऑलरेडी हैं रिपोर्टेड 10 से 11 पीछे एफडीए ने अप्रूव किए हैं. तो नाम मुझे एग्जैक्टली याद नहीं है उनके, बिकॉज इट इज बेस्ड ऑन एंटीबॉडी एंड द ड्रग.
यस सर, थैंक यू सर.
ठीक है. आई होप आई हैव सेटिस्फाइड यू.
यस सर, थैंक यू.
ओके.
Any more questions from audience?
Yes, ma'am. I am having a question. Good
Mm-hmm.
afternoon, sir.
Good
Am I
afternoon.
audible? Sir,
Yes.
like we used to do docking studies. So, if suppose I want to prove inhibitory or matlab excitatory potential of a compound,
Mm-hmm.
so is it possible to prove like in docking? We likely used to prove the binding energies. We can see the binding potential, but through binding we can only see that the particular
Yes, yes,
compound
yes.
is best docked at which.
Molecular docking. मैं समझ गया आपका क्वेश्चन. Molecular docking technique will never tell you whether the ligand is inhibitory, antagonist or agonist. It will just show the binding affinity. ठीक है? तो बाइंडिंग एफिनिटी आएगी,
Okay,
इसलिए
sir.
आप उसको फाइनली इन विट्रो में आपको प्रूव करना पड़ेगा, देखना पड़ेगा. ये बाइंडिंग एफिनिटी आएगी, इंट्रिंसिक एक्टिविटी नहीं आएगी
Okay,
उसमें. ठीक है.
okay, sir. Thank you.
ओके.
Hi.
एनी अदर
Hello.
क्वेश्चन? या.
First of all, it's nice to see you again, sir. I am Farhan Usmani from Uttarakhand. And sir, मेरा
उत्तराखंड.
क्वेश्चन है, अभी मैम ने आपसे क्वेश्चन पूछा कि इनहिबिटरी और एगोनिस्टिक एक्टिविटी के बारे में. तो ऐसा नहीं हो सकता दैट वी कैन कंपेयर स्टैंडर्ड ड्रग और उसके जो इंटरेक्शंस आ रहे हैं, एंड वी कैन कंपेयर जो भी हमारा लिगेंड है उसके इंटरेक्शंस, एंड बेस्ड ऑन दैट
वही करना पड़ेगा ना, वही करना पड़ेगा ना. लेकिन आप कोई नया इनहिबिटर सोचो कि ये जाए, और उसको हम डॉकिंग से ये नहीं पता कर सकते कि जो आपका कंपाउंड है वो इनहिबिटर है या एक्साइटेटर है. ठीक है. क्या कर सकते हैं हम, जैसे ऑलरेडी जो रिपोर्ट अगर इनहिबिटर है उसके बेसिस पे आप ड्रग बना रहे हो तो हमें एज्यूम करना पड़ेगा ये इनहिबिटर बनेगा. बट समटाइम इट हैपेंस, समटाइम सम ड्रग्स कम आउट टू बी एगोनिस्ट ऑल्सो. लेकिन हो सकता है उसकी बाइंडिंग अलग हो, या कोई और अमीनो एसिड्स एंगेज कर रहा हो, या तो जगह अलग हो, या कोई नियर बाय अमीनो एसिड एंगेज कर रहा हो, तो वो एगोनिस्ट भी हो सकता है. मैं आपको बता दूं ना, PKM2 का बड़ा इंटरेस्टिंग केस जो मैंने बताया, वो डाइमर में भी एग्जिस्ट करता है, टेट्रामर में भी एग्जिस्ट करता है. उसमें एक्टिव साइट भी है, उसमें इनहिबिटर साइट भी है. ठीक है. तो वो कुछ जैसे फ्रक्टोज बाइंडिंग साइट थी, जो वहां बाइंड करेंगे वो एक्टिवेट करते हैं, इधर दूसरी जगह करेंगे तो इनहिबिट भी करते हैं, एटीपी बाइंडिंग साइट पे भी इनहिबिट करते हैं. तो इट डिपेंड्स, आपको थोड़ा थोरली स्टडी करना पड़ेगा. लेकिन अगर आप नई प्रोटीन ले रहे हो और आप कोई मॉलिक्यूल डाल के देख रहे हो और फिर कह रहे हो कि ये इनहिबिटर है या एक्सिड, नहीं, ऐसा नहीं पता कर पाएगा. बैकग्राउंड पीछे की नॉलेज तो लेनी पड़ेगी. ठीक है.
थैंक यू सर. थैंक यू.
हाँ हाँ.
Okay, due to limited time frame, I think we should move ahead. And if any participant have any queries, he or she can ask or connect
अच्छा
with.
शिवानी मैडम, आई वुड लाइक टू जस्ट टेल वन थिंग कि
Okay,
जो
sir.
आगे के स्पीकर है ना, दे विल बी टेकिंग केयर ऑफ मॉलिकुलर डायनामिक्स एंड अदर सिमुलेशंस. तो वो थोड़ा अच्छे, डॉक्टर भारतम के ट्रेन स्टूडेंट्स हैं वो. दे विल टेल यू द इन डिटेल कि एमडी क्यों किया, डॉकिंग करने के बाद जब आपने डॉक कर दिया, आपका एक पोज आया, उसके बाद उसका एमडी क्यों किया है, एमएचजी भी ऐसे ऐसे क्यों किया. बिकॉज यू वांट टू सी कि वो ड्रग कितनी देर स्टेबल रहता है, वो कॉम्प्लेक्स. तो एमडी सिमुलेशन 100 नैनोसेकंड, 200 नैनोसेकंड, फिर कैलकुलेशन में बहुत टाइम लगता है, आजकल पेपर में सब मांगते हैं. तो ये आप वो आगे के जो स्पीकर हैं वो बड़े ट्रेंड स्पीकर हैं, एंड आई एम श्योर दे विल सेटिस्फाई ऑल द कमेंट्स. थैंक यू सो मच. सॉरी शिवानी मैडम, आई इंटरप्टेड यू.
No, sir, it's okay. Thank you so much, sir, for this
थैंक यू.
full session. And now it's time to move forward to the next segment of the conference with our next speaker. I think Neha ma'am also connected to us.
Yes,
Ma'am?
yes, I'm here. I'm
Okay.
here.
Am I audible?
Yes.
Go on, please.
Okay, sir. As we move to the next segment of the conference with our next speaker, Dr. Neha Tripathi. Dr. Neha Tripathi is a postdoctoral at GenoScope and Micalis, France. She holds a PhD in pharmacoinformatics from NIPER Mohali and has previously conducted postdoctoral research at the University of Nantes, France, before continuing her work at GenoScope. Dr. Tripathi specializes in computational chemistry, computational biology, and pharmacoinformatics with expertise in molecular docking, dynamics, simulations, virtual screening, machine learning, and deep learning. Her research focuses on drug discovery, structural biology, nanomaterials, biotransformation informatics, leveraging advanced computational techniques to solve complex scientific challenges. She has also gained industrial experience with an AI-based startup in Paris and has made significant contributions to open source drug discovery. As a CSIR and BINC qualified researcher, she has successfully qualified BINC for 5 consecutive years. Dr. Tripathi has published over 20 research articles and played a key role as a co-developer in open source drug discovery initiative, which involved collaborations across 9 national institutes in India. With her rich academic and industrial experience, Dr. Tripathi brings invaluable insights into the field of computational sciences and AI-driven research. Please join me in welcoming Dr. Neha Tripathi. Over to you, ma'am. You can present your screen or you can share your screen for representing.
Share. So, are you able to see my screen?
Yes, ma'am.
Okay, the PowerPoint presentation, right? Is it visible now?
Yes, ma'am, it's visible.
Okay. Okay, I will start. Thank you so much, everyone, for joining and the organizing committee for giving this opportunity. I will try to be brief because I understand, I think we are running a bit late in the conference. I will try to cover this part of the delay. So I will be presenting on the topic computational modeling of targeted drug discovery, insights from molecular dynamics and machine learning. So I have included machine learning really brief. Majority of my research, it comes from computational modeling techniques. I will try to be as general as possible so that everyone can understand this. This is a little bit about my journey, professional journey. So I have an educational background. Hello? I think someone is speaking from behind. This is a little bit about my professional journey. I have an educational background in pharmaceutical sciences, and then I switched my educational field into pharmacoinformatics due to my interest in programming languages mainly. Then I finished my PhD from NIPER Mohali under the supervision of Professor Bhartam. And then I came to France and I stayed here till now. During this period of research, I have mostly concentrated on computer-aided drug design techniques. And recently I have moved to biocatalysis, which I will not be discussing today because it is out of scope for this presentation. This is geographical journey. So I have finished the study from India, especially the PhD from NIPER Mohali, and then I came to France. Currently, I'm employed at GenoScope and INRAE in collaboration, and I will be discussing the results I have generated during my PhD and at CEA and CISAM, because these are related to drug discovery pipeline. These are the eminent scientists which have helped me to gain the insights, deep insights into drug discovery pipeline. And the various disease areas I have been working on include several, but I have been involved mainly with the anti-cancer drug design. So in this presentation, we'll briefly go, really briefly go through the drug discovery and pharmacoinformatics collaboration, because our previous speaker has really entailed everything about this field. Then I will go through even briefer introduction to various molecular modeling techniques. Then I will go to specific results from my research tenure, specifically targeting the target-based drug discovery. And I'm also taking the liberty to show you how computational modeling can be employed to the ecotoxicological evaluation. This I'm going to skip, but I just want to add, in addition to what Dr. Kumar has already said, we also use computer-aided techniques to reduce the attrition rate at the later stages. So we try to remove the harmful or less advantageous compounds at the beginning of the process. This is what I would like to add. I'm going to skip this slide as well, because I think it is very well explained in the previous presentation. I just want to add that a lot of industries are investing in computer modeling. So I think it's evolving really rapidly, specifically the AI and machine learning techniques. This is a visual representation of 3 of the techniques which I will be discussing about. One is molecular docking, where we try to mimic how a small molecule or a ligand is binding to the receptor. In molecular dynamic simulations, because in docking, we consider that everything is rigid or semi-flexible. But in case of molecular dynamic simulations, we try to mimic the physiological environment, because, in our body, we have water. So we try to mimic how a complex that we have generated from the molecular docking will behave when it is put in a bucket of water. Not a bucket here, it's a box, but we try to explore how it will behave in a physiological solution. Then comes the virtual screening, where we try to find the compounds which can have interaction with the receptor similar to what is known for the known drugs. I'm going to skip all the details because you already have it from the previous presentation. I will directly move to the research discussion. So I'm going to start with one of my favorite topic, the drug target in this field, which was also the topic of my PhD research. It's called human topoisomerase 2 alpha. It's an anti-cancer drug target, and it exists in high concentration in the rapidly growing cells, such as tumor cells and the cancer cells. So it makes it a very interesting drug target for this targeting the cancer treatment. In fact, among the currently used chemotherapeutic regimens, at least one drug is present in 50% of the regimens. Now, if you look at it structurally, because as explained before, the structure of a protein is very important when it comes to computer-aided drug design. It's a very complex in terms of structure as well as mechanism of its catalysis enzyme. Now, based on this cycle is really complex. It has many steps, and based on where a particular molecule is acting, it can be classified as catalytic inhibitor or poison. I will not go in details, but this is important for you to remember that there are 2 types of inhibitors, and the poisons, as the name suggests, they are poisons. So they are very potent, but are also harmful. They have more side effects. The second category, catalytic inhibitors, they are a bit less potent, but they have less side effects. So most of the time, combination is used so that we can avoid the horrible side effects from the poisons. Now, I will move to how this actually came into picture in my life. So this was actually an extension of a collaboration from the lab, where our collaborators, who also became my co-supervisor later, they were working on a class of compounds called imidazopyridine and pyrazines. And as the medicinal chemists go ahead, they perform the C2 bi-arylation and C6 arylation of the previously published series, which was published in JMC. So before coming to us, because when you are modifying already existing inhibitors, you expect the compounds to be active in most of the cases and bind at the same binding pocket. So they already did all the in vitro assays where the newly designed compounds were proven to be anti-H2 alpha inhibitors. And also toxic to the cancer cells. Now, the task was very simple. We had to take these compounds, perform molecular docking, provide some interactions, and show that these compounds are binding at the ATP binding site, similar to their parent series. But when we perform molecular docking, you can see here the yellow compounds, they could not fit in the binding pocket. The magenta colored one, which is a natural binding agent for this particular binding pocket. As it is explained before, we go for validation of docking protocol. But in this case, we could not fit the compounds inside. And what you're seeing here, it is almost 60% outside the binding pocket. Now, the question is, if they are inhibitors of the enzyme, why are they not entering the binding pocket? So I suggested this, that these compounds cannot bind at the ATP binding pocket. And our collaborators, they helped to ascertain the hypothesis by performing this ATPase inhibition assay, where you can see that there is not significant difference in the absence and presence of the compound when it comes to the ATPase activity. So this is true. It does not bind in the ATPase domain. But then, if not the ATPase domain, where is it bind? This is the question. So after a very thorough analysis of this entire catalytic cycle, we proposed that these compounds might act at this particular site. It is called Marberon binding site. Now, we have a problem that where do these compounds bind? And we have a solution that they bind at the Marberon binding pocket. But the problem is Marberon binding pocket is not known. So it's our job to identify the Marberon binding pocket and identify how these compounds are acting. Is it similar to Marberon or not? So we did a lot of literature search, which is the best source of any information. We performed molecular docking, proven that these compounds fit perfectly in the Marberon binding pocket. And this identification was actually based on a few known facts, like it's a catalytic inhibitor, it binds near etoposite. So we used a logical reasoning, we performed docking. Now the question is, it is all in silico. So it's true or not, because you can dock anything anywhere, but as it is explained before, the interactions are important. Now, we suggested this to the collaborators and they proposed an assay. It's called attenuation of the etoposide-induced cleavage complex formation. Very fancy name for an assay which shows that these compounds are actually, or Marberon is actually inhibiting the activity of etoposide, which is a poison actually. So interestingly, the new series, which is the series I was working on, showed an effect similar to Marberon, ascertaining what I have proposed, that these compounds actually bind at the Marberon binding pocket. But the previous series did not show any impact similar to Marberon. So thus it ascertained that the previous series was indeed ATPase inhibitor, but the new series is not. Now this publication is a beautiful demonstration of how you can actually change the mode of inhibition for a compound by simply changing some of the structural fragments on the core structure. So this was published in ACS Medicinal Chemistry Letters and was also selected as the cover art for that particular issue. Then we used the complex that we have generated using in silico techniques and subjected it to molecular dynamic simulations, because when you're performing molecular docking, it's very rigid. You don't know how actually the compound is acting in terms of its inhibitory potential. So what we found here, that in the absence of Marberon in the human topoisomerase 2 alpha and DNA complex, you see that the distance between one of the catalytically important residue called as tyrosine 805 and the phosphotyrosyl backbone of DNA, it's around 3.5, which is a good distance to form the intermediate complex called as phosphotyrosyl complex. But when you put Marberon there, this distance is increased to around 4.6 now. So how is Marberon acting? It's actually pulling one of the critical residue towards itself, and it's making it unavailable for the catalytic activity. And that's how it is acting as a catalytic inhibitor. Now we use this generated complex and the understood mechanism of catalytic activity to perform virtual screening, where we did a lot of steps, starting from similarity search in the Zinc database. Now I'm assuming everyone understands these terms because it is already explained before. So the Zinc database, it contains commercially available small molecules. And we used it to evaluate drug likeness. There is a tool called Rulito, which was also developed at Naipur Mohali. Then we performed TopKit analysis to evaluate toxicity potential, ADME analysis to evaluate its ADME property. ADME is absorption, distribution, metabolism, and excretion. And then we performed QuickProp, again, a lot of parameters to see if the compound will perform well at the later stages. Then we went for molecular docking and then molecular dynamic simulations. In the end, we proposed that these compounds, the 6 compounds, have a binding affinity comparable to that of the natural binding agent called merberon. So we can propose that these compounds may have inhibitory potential against the H2O2 alpha. This was published in JMGM in 2019. Now I will move to another example for the same target where we have employed machine learning and deep learning. No, we have employed machine learning only. So what is the basis of machine learning? That you take data, you decide what do you want to predict, then you train your model, you try to validate what you have developed, and then we try to predict the activity that we want to go for. So in this respect, we have actually designed a repository. It's called H2O2MMR. It contains all the compounds which are reported to act on human topoisomerase 2 alpha. And then we developed, we calculated descriptor, we developed a model, we validated the model. It's a very specific process which is well known to anyone who is working on ML. And then we developed a tool called H2PRED. So that's what it's showing. This is the evaluation parameters. This is the model performance, the process. And this is incorporated, the best performing or the top performing models were incorporated in the tool called H2PRED. It's Human Topoisomerase Inhibitor Prediction Tool. This one was published in Molecular Informatics in the year, I think, 2018. So here you can actually predict whether a compound is active or inactive, or you can also predict if a compound is poison or catalytic. So it's a non-linear prediction model. So you can go for this active inactive prediction, and if it is active, you can go for prediction of type of inhibitor, or you can direct, if you already know that your compounds are active, you can go for prediction of whether it's a poison or catalytic inhibitor. Now it's important to have an idea at the beginning. It's possible that it will not be perfect, but it is good to have an idea that these compounds have more potential to be active as compared to the other compounds. So that's where we use it. Pharmacophore-based drug design is also another technique which is widely used. I will not explain this. You already know it from the previous presentation. But what's important here is what we are trying to do. We want a complementary or interacting binding pocket for any compound. So using this, we have proposed potential inhibitors against PLC gamma 1 and SLP76 complex formation. Here we used a pharmacophore based on the structure of SLP76, which is a peptide. And this is an interesting approach because it's not easy to design inhibitors against this PLCG gamma 1 because there is no specific binding site. So this can be actually considered as undruggable targets. So it's very difficult to design something against these targets. There is another report using the similar approach. It's called, the inhibitors of CD2 and CD58 interactions. This was designed as immunomodulators, and the first one was anti-cancer drug design. Now, this one is, again, we used molecular docking-based virtual screening to identify potential inhibitors against hepcidin, which is a biomarker in the anemia of inflammation. So the people who have like rheumatoid arthritis, if some of you know some people who have rheumatoid arthritis or any inflammatory disorders, they tend to develop anemia in future. And the culprit is this hepcidin. So its concentration in blood is increased. And what it does is it goes and binds to the ferroportin, which is responsible for iron absorption, and it destroys it. It actually induces the decomposition or degradation of ferroportin. And now you don't have the transporter for iron, so you don't absorb iron and you develop anemia. So the idea behind this project was to design some compounds which can actually engage hepcidin. It can take them to a trip, and so that hepcidin will forget its function of destroying ferroportin. In this process, we found some potential inhibitors, and this was also proven in vitro in mice. The poor mice developed anemia when it was induced with the hepcidin, and the compounds that we discovered helped to reduce that anemia. This work was published in scientific reports in 2017. Now, most of you are, if you are from pharmaceutical background, you are aware of the term called polypharmacology. So in polypharmacology, we try to design or identify multiple targets for the same drug. This is it is of course applicable to a lot of disease areas, but I have worked mainly on the anti-cancer drugs who have multiple drug targets. And the drugs that I have targeted on, it's quinacrine, etoposide, and doxorubicin. You must be hearing etoposide a lot. You heard it before also, but now you will be hearing quinacrine a lot. So what we did is we actually tried to evaluate the activity of these three compounds on the DR5 trail axis. Now, what is so interesting about this project is this is one of its kind. So in this project, we, for the first time, identified a binding pocket for the compounds who actually affect this particular axis. And another interesting thing here is these compounds were actually agonists. So as already explained to you before, it's not easy to tell you whether a compound that you are trying to find is an agonist or antagonist. So I will explain how a carefully designed protocol can actually tell you whether the compound that you are working on will act as an antagonist or an agonist. So what we did here, in this case, we want to target the DR5 trail complex because it has a high concentration in the cancer cells. We don't want to target DR4, which is universally present in all the cells in the same concentration. So we want to avoid the side effects as much as possible. What we did, we took quinacrine, etoposide, and doxorubicin as the positive control. Like any experimental stuff, you need a positive control, you need a negative control. So we took dexamethasone and piftrine alpha as negative control because it is known from experimental studies that these compounds do not impact DR5 trail axis. So what we did is first, we identified a binding pocket at the interface of these two macromolecules. That is the one in cyan color or the blue one, this is called DR5, and the one in green is trail. Trail is the ligand, DR5 is the receptor. And in general, there is anti-cancer antibody. It's called AM655. It is a approved antibody against this axis. So what it does is what you're seeing in the graph, the green colored one, it actually increases the binding affinity of the two complexes, this complex, the DR5 and trail, as compared to the binary complex that is in red in this graph. So you're seeing that such a big molecule, the antibody is really big. It is increasing the binding affinity by almost, I would say, more than 40 kilocalorie per mole. When we put quinacrine, etoposide, and doxorubicin, we noticed that this small, really small molecules as compared to the antibodies, they increase this binding affinity by almost like 4 times, I would say, 3 times at least. What is another interesting fact? That the binding site that we identified, it's all in silico, you can put a compound anywhere, you will find the interaction at least 1 or 2. But then it was proven in vitro that the binding site that we have identified is indeed the binding site for these anti-cancer agents, because when they mutated this CD68 to alanine, these compounds could not interact experimentally with the DR5 trail axis. So this work was published in 2 parts, once for quinacrine and then for etoposide and doxorubicin in oncotarget apoptosis. And this is one of its kind for, that's what I was saying, the FAB, the antibody is really big. This, what you're seeing here, the cyan and magenta is the antibody. So we are able to replace, and there are problems because antibodies are proteins, their delivery, bioavailability, there are so many issues. So actually you can replace them with small molecules. So this work has opened a new paradigm for DR5 trail agonist design. So this agonist term is important here because it's the first time I think someone has reported a design of agonistic ligands, if I haven't missed some of the articles. Another study, it's again concentrated on quinacrine. So our collaborators, they came to us and they wanted to know if how this GLI1 and DNA interaction is inhibited by this quinacrine. So what we did is, again, we designed a protocol because GLI1 is an untriggable target. It doesn't have a binding site where a compound can bind. But quinacrine is known as a DNA intercalator and it binds between GC-GC base pairs. I'm assuming everyone knows DNA here. So what we did is we performed the intercalation of quinacrine because you can intercalate, but then it has, everything else has to adjust in the presence of the drug. Then we perform molecular dynamic simulations. As I explained to you, molecular dynamic simulations can show you how your complex is behaving under dynamic condition. So what we observed here, the blue color here indicates positive charge, the red color indicates negative charge. And we all know the DNA backbone is negatively charged. Now, in a binary complex, when you don't have quinacrine here, you have DNA backbone, which is red, negatively charged, interacting very well with the positive charge of the GLI1. But when you put quinine in this place, there is a distortion in the DNA, which is leading to a structural disturbance in GLI1. And you see the negatively charged DNA backbone comes in contact with the GLI1 red part, which is electronegative. You can see that the binding affinity is almost reduced by half here, a little less than half, in the presence of quinine. And that's how probably it's inhibiting, or actually it's throwing away GLI1. So quinine is acting as a second partner, which is throwing away the first partner out of the picture. This work was published in Scientific Reports in 2016. And again, it's the first of its kind, where we have used molecular modeling techniques to identify novel binding pockets. Now, this is another example where it's not related directly to drug design, but here I just want to show how beautifully the molecular modeling techniques are able to show you that the single mutation, although these 4 peptides come from Huntington's peptide, and out of this, the 3 are mutants. The position of them is shown here, the AAA alanine. What we observed, not we, our collaborators observed that out of these 3, one is behaving differently as compared to the other 2. And usually you will assume that it shouldn't impact so much. So it was behaving structurally different and functionally different. It was behaving functionally different experimentally. So we try to evaluate why it is doing that. So we perform molecular dynamic simulations after mutation. Again, the dynamic simulations will try to adjust the structural parameters due to the mutation. We observed that what you're seeing here in magenta in these 4 graphs, these are secondary structure graphs. The magenta shows the alpha helix, but the last one you will see a lot of green color, which is actually showing that the alpha helix is broken. That could be the reason why it is not able to induce the aggregation of protein in experimental situation. So this was again, it was published in Feb's journal in 2018. This is the first paper from my entire research career, and it's from my master's in 2011 when I finished it. So here we have shown for the first time how this the GABA A receptor is actually opening and closing. So it's something like a bottle opening and closing process. So it's again first report. So there is a leucine ring in the middle of this channel, which actually form a closed interaction with other 4 leucine and closes the channel. And when the channel is opening, it rotates to increase the distance between the leucines to open the channel. And that's how it's allowing the flow of chloride ions in the ion channel. It is a very important drug target in anti-epileptic drugs design. So employing molecular docking, and then subsequently you can use this information to see if your drug will act as an inhibitor for this channel or not. So this is another pioneering work from my research career. Now I will show you something related to ecotoxicology that comes from plastics, and I really want to share this because we all know how bad plastics are. So we are eating plastic and we are spreading plastic everywhere. It's getting incorporated in the ecosystem very, for infinite times. So based on the size of your plastic, you can classify it as microplastic or nanoplastics. And during COVID, it was reported that around 26 tons of healthcare waste reached ocean. Out of the 12 tons of this plastic was actually micro and nanoplastics, which cannot be recycled. And that's the bad thing, because if you cannot recycle, that means it is in your ecosystem. And what happens, even if it is there, let it be like everything else. But it attaches to the biomolecules and it distorts their function by distorting their structure, disrupting their structure. So there are a lot of studies already which have reported that the binding of any protein molecule to a plastic can actually affect its structure and function. As you can see in this figure, this yellow thing, it's insulin. So binding to a plastic surface is causing insulin fibrillation. Similarly, although it is assumed that the fetus is safe inside a mother, but it has been reported that these plastics are able to transport, to actually cross the placental barrier and reach the fetal blood through mother. And this was proven in vivo using a mice. When a pregnant mice was given was made to inhale a plastic-loaded air, and the fetus was affected. Again, the poor mice was exposed to water loaded with plastic, and it reached the brain. So probably we all have plastic in our brain. We are plastic people. Now, what we did here was we tried to generate the plastic nanoparticles using polyethylene and its oxidized counterpart. We placed them in 3D, subjected to simulated annealing. And when you perform simulated annealing, they fold. So we performed 2 cycles, and it was a very stable globular nanoparticle. We have 2 types of nanoparticle. What was interesting here, that this nanoparticle, the filaments tried to actually arrange themselves in parallel to each other to have less frictional forces and have a smoothly sliding filaments. Now, what we did is we used this to create larger nanoparticles, and what I will show in next slides is the one which is named as 27NP. So it was generated after joining 27, this smaller globular particle. That's the name. Now, the protein we have selected is called human alpha-synuclein, which is a very important biomarker in the Parkinson's disease. I hope everyone knows what's Parkinson's disease, but what's important here is that this protein is very disordered structure. And what's important, another important thing is this part, the helical structure, is the pharmacologically important part of this protein. What it does, it does something in the neurons, and it actually facilitate the release of neurotransmitters. So now if your alpha-synuclein is not working, you will not have neurotransmitters released, and that's the sign of Parkinson's. What we did is we took the protein, performed molecular dynamic simulations, obtained a compact form because it's a very complementary structure, electrostatically complementary protein. So it actually became very compact. We took this, placed in the vicinity of our generated nanoparticles, then we try to evaluate whether they will interact or not. We have 2 types of plastics, but I will be showing you results of non-oxidized one only, the one without red color, because the one with red color, the oxidized one, it didn't interact with our protein. So we took this, we performed simulation, and we observed a rapid anchoring and absorption of alpha-synuclein on the surface of nanoplastic. And this was also proven using the experimental techniques where the nanoplastic and alpha-synuclein, they form a very stable complex. And what is interesting here is the interaction occurred through this helical region, which is the pharmacologically important region of this helix, of this protein. When we tried to put more proteins in the vicinity of nanoplastic, we observed that anything that you put will go and get stuck on the plastic surface, except for this one, but I will explain why it happened. So now we have like 3 completely absorbed alpha-synucleins, and 1 is hanging on the surface. But the ones which are completely absorbed always bind from the same side, and it's irrespective of their initial orientation. Anyway you put it in the vicinity, it will always go and interact to the same surface. And the interaction surface area of this protein was calculated in silico and was found to be equivalent to what was calculated experimentally, which is ascertaining our findings from in silico based on the experimental results. What is interesting to see, wow, even if it got absorbed, what's the problem? Let it be sitting on the surface of microplastic. But what we observed, that the green one you are seeing here is the one which was in complex with microplastic, superimposed with the unbound form, the red and the blue one. You will see that there is a kink in the helix, that it's a bit bent. And because of this bending, it cannot perform its pharmacological function. Now we observed that the hanging human alpha-synuclein that you observed, the red colored one, was having very strong interactions with the one of the alpha-synuclein on the surface. And why it happened? Why it cannot happen when the protein is in water? Because the C-terminal domain, which is a disordered region, is a very dynamic structure, disordered structure. So it is, when somebody tries to grab it, it moves away. So you need it to be more immobilized on the surface of nanoplastic, and that's how the other alpha-synuclein is able to interact. And this was proven using experimental techniques where the protein aggregation was observed in the presence of nanoplastic, but not in the absence of. So this is another example where experimental techniques have confirmed the findings from computational studies. Now, so I would like to conclude here for this part, and then I will conclude for the next part. Here we have a very strong interaction in the non-oxidized polyethylene plastic, and that's why the government is asking everyone to not throw the plastic particles in open and recycle it responsibly. So when it binds, it is unavailable to perform the neurotransmitter release, and that's how people get Parkinson's, and that could be the reason why there are increasing incidences of neuronal disease. So this is a list of my publications, but that's not what I want to show here. What I want to show here, that the one in green and the blue, they are in collaboration with experimental studies. So what you understand here from here is that if you design carefully, it's possible to obtain really interesting results from computational studies and have important contribution to the discovery pipeline. So I would like to conclude my presentation by saying that this computer-aided drug design is really rapidly evolving. In addition to the molecular modeling techniques that we have already seen, nowadays ML and DL are becoming really, really relevant. And my current project, it completely concentrates on DL, and transformers is a term that if someone is trying to enter into DL, they should know, because it's really revolving. We have recently submitted a review on application of DL in biocatalysis. So a lot of companies, they are investing in new approach methodologies where you can prevent killing of poor mice as much as possible. So that's how a careful design of protocol is important in molecular modeling techniques. So I would like to acknowledge a lot of people, but especially the organizers and the contacted person, Chetna, thank you. And audiences for your patience. And I'm sorry if it was too much to grasp in 1 hour. And this is just a little bit about life in France. So because it's part of Europe, so you get to have a lot of exposure to the scientific meetings around. So I have visited Sweden for one of the conference, and similarly to Grenoble, and these are all parts of France and Europe, other countries. Thank you so much, and I will be happy to answer questions as much as possible. So I will stop sharing on this. Okay. Okay, so if there are any questions.
Thank you, ma'am, for delivering such a thought-provoking and informative session. We sincerely appreciate your time and effort in sharing your knowledge with us today. It's a time for our discussion and question answer round. So let us begin. Who want to ask any questions? Audience? Any audience who want to ask any questions?
Hello, ma'am. This side Pooja. Okay.
Hi, Pooja.
Yes. You're this. I was just listening. It was so interesting, and you made it very easy. The plastic, how it at the molecular level, with the pictures and with the presentation was very good. And right now I'm in Punjab. I think you have done it from Naipur Mohali, Punjab only.
Sorry, about the plastic
PhD.
PhD I have done it from Naipur Mohali, and the plastic thing that you have seen, it's done from France. So I think yes.
Okay. It was very appreciating. And your domain is computational chemistry. And I would like to join you. I would like to work with you if you agree, or a small part I would like to share, or in future, or after I'm doing my PhD and I'm approaching for the postdoc also. Are there any opportunities for postdoc in your area, or like
yes,
that? Can you guide regarding this?
There are indeed a lot. I can share. I have recently received some of the listings. I can share them.
I would like to get your mail ID, or how I can connect to you.
Okay, maybe one of the organizers can share it. I don't have a problem. I mean, I can put it here, but I don't know if it's a good idea or not. Sorry?
If you give us content, we will share your contact with the audience.
Okay, yeah. I can, probably Chetna can share if she has, it's okay?
Okay, ma'am.
I have no problem. I will be more than happy to help as much as I can. Yes.
Thank you so much, ma'am. Yes.
All the best.
Yeah.
Okay. Is there any other question?
From audience, any questions? Anyone who want to ask the questions?
Good afternoon, ma'am. Yes, ma'am, I have
afternoon.
a question, ma'am.
Tell me. Yes. Hi,
Yes,
Shivansh.
ma'am, my question was, hello, ma'am. My question was regarding the Parkinson's disease and the nanoplastics. Does the nanoplastic particles that you showed, does they act as the nucleation sites for the protein aggregation?
Yes, exactly. They act as nucleation site. I didn't want to use term. I wasn't sure if everyone would be aware.
Yes, ma'am.
So yes, we have recently published this article. It came online just last week. It acts
Yes,
as a
ma'am.
nucleation site, and in fact, it acts as a nucleation site, of course, in silico, as well as in experimental conditions as well. So we have proven it using, not me, my collaborators have proven it experimentally, that indeed, when you put the microplastics and protein, you observe protein aggregation because it's immobilizing the C-terminal domain. It's allowing the other protein molecules to form interactions with already absorbed ones. And if you don't put microplastics around, you don't see that kind of aggregation. So yes, it acts as a nucleation site.
Okay, ma'am. Ma'am,
Hello,
one more
ma'am.
question.
Yes. Yeah. Can I ask one question?
Yes.
This microplastics, can we apply it in a positively in the drug discovery in any other way?
It's not the microplastics that you can apply because you don't want to put microplastics in your body intentionally, but the similar approach is being used for other nanomaterials, which are very important for drug discovery. For example, there is something called immobolites. It has similar structure, and there is a difference in the microplastics and other nanoparticles because it's a soft matter. The microplastic you have seen, they are moving a lot. They don't have a crystallized structure as such, although there was a crystallization induced when you put the protein around. But you can use the same approach for other nanomaterials because it's assembly study. So most of the self-assembling nano drugs, if you forget that the particle that I have shown is a plastic, you can use the similar approach to evaluate other nanoparticles, but don't put polyethylene nanoplastics as drug in a body. You don't use that as a drug, but you can use the same approach
In delivery,
to apply
yeah,
to
in
the
the delivery, yeah.
Yes.
Novel
I
delivery.
think
Good afternoon, ma'am.
Good afternoon.
Ma'am, I want to ask, can you share that slide of the publications again?
Yes, I can send it to organizers, and I don't have a problem.
Thank you so much, ma'am. Please do share your contact to contact you by email or somehow.
Okay, or also, I mean, the complete list is also on the Google Scholar, Neha Tripathi. So I can share the slide as well as this, no problem. Shivansh and Miss Simran Jot. Yes, someone has a question, I think.
I have asked, ma'am. Simran.
Yes, ma'am, I had one more question regarding the nanoplastic only. Ma'am, like, what are the potential transportation mechanism of the nanoparticles? Because it is very difficult to cross the blood-brain barrier. And once it's like, there's one add-on question to it, like when it crosses the blood-brain barrier, so like, does it integrate any other brain susceptibility to more toxins, like nanoparticles and other toxins?
I am not sure if I understood your question clearly, but the slide that I was showing before, there, let me share. So here there was an in silico study for the transport stuff. Sorry. Okay. Shivansh, so this is the study. What they have done is it's a passive transport through the blood-brain barrier or any membrane. Actually, it's highly hydrophobic, your nanoplastics, most of the time, especially the polyethylene ones. And you have a membrane, it's also highly hydrophobic. So people have actually demonstrated it in silico for sure, that it actually integrates in the membrane and then it passes through the other side. So it has been known. I am not aware actually if there is any other way, but it's already known that it enters. So there is a mechanism to transport, because you see it can cross the placental barrier. We know there are a lot of insulin problems, diabetes, so definitely it's going to pancreas as well. It's going to brain for sure, because this was a very stressful publication. It was recent, I think, and they were not too recent, but like 5 years ago. And there was like really hot topic. It became a hot topic because everyone was worried about, oh, my brain has plastic. So I think it's well known. So most of the time it's passive, because nanoplastics are mostly hydrophobic. They can easily integrate your membranes. So I don't think any channel is involved. I said most of the channels are working with ions and charged stuff, but there are some plastics which are charged also. So anionic plastics and but I have skipped that part. But most of the hydrophobic plastics, they are less harmful, because in fact the plastic discovery, it's being targeted on designing the hydrophilic ones, because they are less harmful, because they have a very strong water shell around them. And it's very difficult for any protein or biological molecules to replace this water shell. I shouldn't have deleted the slide. I didn't expect this question, but yes, the water shell is so strong, and we observed that with time, the number of water molecules forming hydrogen bonds with the surface of microplastics, it was increasing. And now, hydrogen bonds, they have like 2 kilocalories for sure, and there were like 500, hydrogen bonds. So it's so difficult for this alpha-synuclein to replace it, and it would be true for any protein. So the hydrophilic ones, they don't cause any problem in the body. It's very difficult for them to transfer. So I think the hydrophobic ones are only working. The transport mechanism should be through the membranes to passive transport. That's what I think comes from logical reasoning, but there can be something else I have to look into.
Okay, ma'am. And there was this add-on question to it as well. That does it integrate the susceptibility of blood-brain barrier for the other environmental toxins as well? Like once
I don't think. Yeah, I understand what you mean, but I don't think it damages the blood-brain barrier. I don't think it damages it, but it crosses it, and then it lets the blood-brain barrier work on its own. But it can act as a nucleation site for other toxins and take them along with it, like a camouflage. So other toxins which can actually not cross the blood-brain barrier may cross when they are on the surface of microplastics. So it is not as such damaging the blood-brain barrier, but it is letting something which should not cross, crossing with itself. It has a ticket to go through blood-brain barrier because of its hydrophobicity, but then it attaches something which shouldn't be crossing, and it can go through the blood-brain barrier. So it doesn't damage, but it does cancels the purpose. So I think.
Thank you so much, ma'am.
Thank you so much, Neha ma'am, for your valuable presentation, and it is very insightful presentation, ma'am. Thank you so much.
Thank you, everyone. You're welcome.
Dear participants, all the feedback form links has been shared in the chat box. Kindly fill it out. And now there will be a virtual lunch break from 1 to 2 p.m. and all the participants are requested to rejoin at 2 p.m. for the next segment of the event. Thank you so much, everyone.